All right. So today we are going to talk about metaphase in this part of the chapter. And that includes prophase, metaphase, anaphase, telophase, and finally cytokinesis. First, we're going to watch a short video about this process, and then we'll go on to talk about it.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation. Mitosis. In this
All right. So in the video, you saw prophase, anaphase. Prophase, metaphase, telophase, or anaphase, telophase, and cytokinesis. First, we're going to talk about prophase, and this is where the chromosomes become visible. They have replicated each other, and there are two chromatids joined together at that centromere. The chromosomes then separate and migrate towards opposite poles, and those mitotic spindles and asteres form. So they are starting to double and move together towards that center on opposite sides of each other. Then the nuclear envelope fragments and the microtubules attach to the centromeres and draw them towards that equator or that middle of the cell. And the polar microtubules assist in forcing those poles apart. So we can see early prophase here, everything separating and moving to opposite sides of the cell. And those chromatids have duplicated, and now you can see that traditional shape. The late prophase, you can see that they're separating themselves on the polar sphere and coming together, and that nuclear envelope has dissolved or fragmented. Metaphase is where the centromeres of the chromosomes are aligned, so there is a nice line through the middle of the cell, and the plane midway between those, or that nice line, is called the metaphase plate. So we can see the metaphase plate here at that line. We can also see it here on this fluorescent microscope image. You can see those spindles as well and the chromatids showing up very nicely. Anaphase is the shortest phase, and this is where the centromeres of the chromosomes split simultaneously, and each chromatid now becomes its own chromosome. These chromosomes are now V-shaped and are pulled towards the opposite pole by motor proteins and polar microtubules pulling them completely apart. So you can see they've separated at that plate and are now pulling apart. Telophase begins right at the end of anaphase, and this is part of cytokinesis. And it begins when the chromosome movement stops. The two sets of chromosomes then begin to uncoil and form chromatin. The nuclear membrane then forms around each chromatin mass, and the nucleoli reappear. Those mitotic spindle fibers that have been pulling the chromosomes apart then disappear. Then cytokinesis begins in that late anaphase, right before telophase, and a ring of actin microfilament up here. It's kind of like I'm so sorry. A rubber band around something, and it starts to slowly constrict, pinching the two daughter cells apart, containing two. Each daughter cell then now contains two nucleuses with identical material in them. So telophase and cytokinesis, we see that ring of cleavage furrow show up, and it's going to constrict to make those two cells independent of each other. So what controls our cell division stage? So we have two different types of signals. We have go signals, which tell cells to divide, and we have stop signals. So go signals are super important. So they tell cells to divide. If you have an injury, it'll tell your cells that they need to go and produce more cells in that area to fix that wound. Or if you're not absorbing enough nutrients, let's go add some cells there to increase that membrane exchange in a hypertrophy way. So critical cell volume of cells will trigger a go signal, and those go signals can come in forms of chemicals like growth hormone factors, hormone cyclins, and cyclin dependent kinesis, or CDKs. And these hormones will then go tell the cells will take them in, and then go tell the nucleus that it's time to divide. We do have our stop signals, and stop signals, we have two different ones. We have contact inhibition. So if there are too many cells in an area, they will not signal each other to divide. Actually tell them other cells around them not to divide. It's like too many people in a room. Hey, we can't fit any more people in here. Let's not get any more people in there. Kind of like the buses on field day this year. We couldn't send any more kids on the bus, so we were all smushed together. that would be a stop signal. Growth inhibiting factors also produce repressor genes. So they produce, growth repressor genes will produce growth inhibiting factors to tell cells not to divide and signal cells around them not to divide either. So now that we've talked about mitosis and DNA replication, let's talk about protein synthesis. Protein synthesis is super important because proteins do 90% of the things in our body. So how do we make proteins to make our body do things, right? We talked about proteins just even in mitosis and DNA replication. So we need proteins doing everything else in our body. So DNA is the master blueprint for protein synthesis. DNA tells the cells what proteins to make, and that the structure of the protein then dictates the function of the protein. So a gene is a segment of DNA that blueprints for one polypeptide. So a gene makes one protein, one type of protein. And each one of those genes has something called a triplet code. And these triplet codes are nucleotide bases formed from genetic libraries. And each triplet is specific coding for an amino acid. Now, if we think back to biotechnology and MedMicro, amino acids are the monomers of polypeptides or proteins, the polymers. So amino acids will link together in a peptide bond formation to make polypeptides, which will then fold and make proteins. So it's super important to know that each triplet code from the DNA will make an amino acid. Those amino acids will link together to form a polypeptide, and those polypeptides will form to make a protein. So we have two steps to make proteins. We have transcription, which takes place within the nucleus, and that takes DNA and turns it into mRNA. And then we have translation, which translates the mRNA to make amino acids in a polypeptide chain. So mRNA, we do have three different types of mRNA or RNA that all contribute to the steps in translation and transcription. mRNA is the first one we're going to talk about, and it carries the instructions for the building blocks of polypeptides from the genes in the DNA to the ribosomes in the cytoplasm. So it is our messenger. It literally carries the message from the DNA out into the cell to make that protein. And they have the triplet code on them. Then we have ribosomal RNA, and these ribosomal RNAs are structural components of a ribosome that move along the mRNA to translate the message. So it is the housing block to move along to translate mRNA. And then tRNA is transfer RNA, which is the amino acids that bind with the base pair codons of the mRNA that then produce an amino acid. So they transfer the amino acid to the polypeptide chain in the sequence that the mRNA or the message gives it, that rRNA or that ribosomal RNA houses the transmission of information to happen. So let's walk through the complete steps of transcription and translation, start to finish. So transcription transfers DNA genes based on a sequence to a complementary base sequence of mRNA. So there are some factors that have to happen. We have to loosen those histones that DNA is wrapped around in the nucleus in order for it to be transcribed. Then we have to bind a promoter, which is a protein, to the DNA sequence, specifically at the start site where the gene is going to be transcribed, and then it's going to mediate the binding of RNA polymerase to the promoter. RNA polymerase is an enzyme that oversees the synthesis of mRNA, just like DNA polymerase. It unwinds that DNA template strand, adds complementary RNA nucleotides to the DNA template, and joins them together. It stops when it reaches a termination signal, and that is just a signal that tells the RNA polymerase that this is the end and we need to move on. mRNA then pulls off the DNA template strand and is further processed by enzymes before it enters the cytosol, or the cytoplasm. So here's a nice little fancy image for you. We can see that promoter region comes along and RNA polymerase will bind to it. Then RNA polymerase is going to move along and start transcribing mRNA. And mRNA is then going to elongate. So we have the initiation, right? RNA initiates transcription. And then it elongates the mRNA. And then it stops at the end when it hits a termination signal. So then it's termination. So we have 3 steps: initiation, elongation, and termination. And then RNA polymerase removes itself, mRNA detaches itself, and then moves over to an enzyme to be processed. So, like I said, here's a close-up look at what this is doing. So, RNA binds to the promoter region with the help of transcription factors, and it pries the 2 DNA strands apart and initiates mRNA synthesis from the start point. Elongation, as the RNA polymerase moves along the template strand, the elongation of mRNA happens 1 base at a time as it unwinds the DNA helix and rewinds it behind it. And then termination, it gets to the end over here and it pops off and mRNA removes itself. So the DNA RNA hybrid, so right here, the hybrid region, is between 16 to 18 pairs at any time of DNA and RNA is still bound to DNA. So where the DNA and RNA are bound together, this is called the hybrid region, and it's about 16 to 18 pairs. Here's the overview again. So then we move over to translation, and this is where we convert the base sequence of nucleic acids from mRNA into an amino acid sequence of proteins. So it translates from nucleic acid to amino acid, and this involves all 3 types of RNA. So each 3 base pair sequences on DNA is represented by a codon. And a codon is a complementary 3 base sequence of mRNA. So each 3 base sequences of mRNA is going to have a complementary codon. So this is the chart we use for our 3 base pairs. So base 1 is the first letter, U, second letter, U, third letter, U. And we can figure out which amino acid it is. So what happens in this? mRNA attaches to a small ribosomal subunit that moves along the mRNA to the start codon. The large ribosomal unit attaches and forms a functional ribosome. These are our 30 and 60 subunits, if I am thinking of human cells. Then we have an anticodon of tRNA, bind to the complementary codon and add the amino acid to form a protein chain. The new amino acids are added one by one from another RNA as the ribosome moves along the mRNA until the stop codon is reached. So here's a nice big overview, and we'll talk about each step individually. So after mRNA is synthesized, it is processed, and mRNA leaves the nucleus and attaches to a ribosome. So it is processed first, and then it leaves. Our tRNA, while this is all happening, is going to go off and find an amino acid, and it's going to pair together using ATP. So that mRNA is going to be bound into our ribosomal or our RNA. We have our large subunit on the top and our small subunit on the bottom, and it's going to go, once again, 5 to 3. It is a directional ribosome. It is read directionally. Each ribosomal large subunit has 3 sites: the E site, the P site, and the A site. So this RNA is already transcribed over here, and this is what's getting transcribed. So translation begins when an oncoming tRNA recognizes a complementary codon and binds to the A, or the accepting site, in hydrogen bonds. Then the ribosome moves along the mRNA, and each codon is read. The new amino, so this tRNA moves here, and it then moves to the P site, where it will add its amino acid to the amino acid chain. Then, as the ribosome moves along, the P site moves to the E site, and the E site, I remember what the E site does because it ejects the tRNA, and then continues onwards and so forth. It releases the tRNA to go find a new amino acid to add to that strand. So there you go. You can see that. So what is the role of the rough ER in all of this protein synthesis? So mRNA, the ribosome complex, is directed to the rough ER using a signal recognition particle. So it's just a little chemical particle that says, hey, come here. And all of this that we have talked about, the translation, typically happens in the ER. So sugar groups may be added to the protein, and its shape may be altered as well in the ER because they have all of those ribosomes since they're studied. Proteins then are enclosed into a vesicle and then transported to the Golgi apparatus once they leave the rough ER. So we can see here, the signal is recognized, transcription happens, the growth of the polypeptide chain, it is removed, sugar groups are added, and then it moves out into a transport vesicle and moves over to the Golgi apparatus. So, step by step, mRNA ribosome complex is directed to the rough ER by the SPR, and the SPR binds to the receptor site. So this is the rough ER, this is where it's happening. Once attached to the ER, the SPR is released, and the growing polypeptide chain snakes through the rough ER membrane, pour into the cysteine. The signal sequence is clipped off by the enzyme, and a protein synthesis continues where sugar groups may be added to the protein, changing its structure, altering its function. Finally, it's released and moved over into the vesicle, where it's pinched off and transported to the Golgi apparatus. So other roles of DNA. So I said it had to be processed before it moved out of the nucleus. And that's what happens. They remove the introns, or the junk regions of the DNA that code for other types of RNA that is not needed in that DNA synthesis. We also have antisense RNA, which prevents protein coding RNA to be translated. MicroRNA, which is small RNA that interferes with mRNA made by certain exons. And then we have a riboswitch, where a folding RNA acts as switches that regulate protein synthesis in response to an environmental condition. So antisense prevents coding from happening. MicroRNA is just interfering, and riboswitches switches on and off protein functions. And those are all introns, or junk regions. All right, we will end here for today. We will pick up here tomorrow. Y'all have a great day.